All right, so this is going to be the last step of cellular respiration. This is where we're going to produce the most ATP. It is a little bit confusing because there's kind of like two separate things going on here, but it's very similar to the electron transport chain that we saw in photosynthesis in the light dependent reactions. So we have seen some of this stuff before in a sense. So this is going to occur along the inner mitochondrial membrane. We're going to be looking at that inner membrane space as well. But the membrane proteins here on the electron transport chain are going to be on the inner mitochondrial membrane. And if you remember that cristae, those folds in the inner membrane are going to increase the surface area there. That's going to increase the amount of these membrane proteins and the enzyme ATP synthase, which is essentially going to increase the efficiency of mitochondria. We want to be able to break down glucose as fast as we can, especially during times of like fight or flight, where your body is in a like critical moment where you have to fight somebody off or you have to leave. So looking at this first step here, NADH and FADH2, we talked about how these molecules are carriers for electrons. Remember, these are like the taxis, like Uber and Lyft. They're going to take people from one place to the next. They're going to take those electrons and the hydrogen ions to the electron transport chain. What they're going to do with the electrons is they're going to donate those electrons to these membrane proteins, and they're going to drop off these hydrogen ions. You can see here these H pluses. Remember, they're just protons, but we signify them as hydrogen ions. It's a little bit easier for us to just identify them as such. Now, just like in the electron transport chain, these membrane proteins are going to use the energy in the electrons, and they're going to pump hydrogen ions into the inner membrane space. That's going to come next, though. All right, now that we have this concentration gradient, they're going to start moving, these hydrogen ions are going to start moving from areas of high concentration to areas of low concentration. You can see here we have ATP synthase again. Again, this is my favorite enzyme. It's just so cool looking. It's just beautiful to me. But this hydrogen ion is going to go through this ATP synthase, and it's going to rotate it just like it did in photosynthesis. Nothing really new here. All I need you to understand here is that as this rotates, because the hydrogen ions are moving from higher areas of concentration to low, this thing's going to rotate and bond a phosphate group to a DP. Now, we talked about substrate level phosphorylation back in, I think, the first section or first or second section. That's when you take a enzyme and you take a substrate. So you take the phosphate group off the substrate and you attach it to the ADP molecule. That's different than what's going to go on here. So substrate level phosphorylation only accounts for the 2 ATP produced in glycolysis and the 2 ATP produced in the Krebs cycle. In the electron transport chain, this is oxidative phosphorylation. This is when we make ATP by the movement of electrons throughout the electron transport chain, which is coupled with the hydrogen ions moving through the ATP synthase here. Again, this produces 90% of our ATP. We're going to get a lot more ATP here per glucose molecule than in the substrate level phosphorylation. This is, again, where we produce a majority of the ATP in the electron transport chain. So as you can see here, the electrons are going to move down through this electron transport chain. Please don't memorize any of these membrane proteins. There are a bunch of membrane proteins here, but they're going to donate, or they're going to push those hydrogen ions to the inner membrane space here, and they're going to create that concentration gradient. There's going to be a high concentration of hydrogen ions in the inner membrane space and a lower concentration in the mitochondrial matrix. That's going to be exactly like what we saw in the thylakoids in the chloroplast. There's going to be a high concentration inside the thylakoids than outside. And specifically, if you remember this inner membrane space, there's not a lot of room here. It's just the outer portion of the mitochondria. Again, we're reducing the surface area to make it easier for us to create that concentration gradient. This is another great image of what's going to go on here. You can see the NADH and FADH2s. They're going to donate those electrons to the electron transport chain. Along the electron transport chain, they're going to move those hydrogen ions into the inner membrane space. You can see there's not much room here in the inner membrane space, so it's pretty easy for us to move those hydrogen or to get a high concentration of hydrogen ions here and a low in the matrix. Now, I haven't gone over this yet, but I want to start by saying those electrons, the final donator is going to be oxygen. So those oxygen molecules that you have in your mitochondria are going to bond to the electrons and some of the hydrogen ions in the matrix, and they're going to form water. If you remember, The first step of photosynthesis was what? It was water breaking. Yeah. The last step here is water being formed. Now, it's not like this reaction just occurs flip flop and totally backwards. No, but we do see similarities between photosynthesis and cellular respiration. And this is one of them is the first step in photosynthesis was water breaking. Now we have oxygen forming into water here instead of the opposite reaction. Now, I'll talk about this a little bit later, but just like in plants with photosynthesis, if you don't have water, nothing else works. If you don't have oxygen, everything else gets backed up, and I'll describe that later. Now, again, we have these hydrogen ions that are going to move through. Again, we have a high concentration of these hydrogen ions. They're going to move through the ATP synthase. As it moves through, that's going to power it to make it swivel like a propeller, and that's going to bond the ADP with a phosphate group to make it ATP. Now, remember, this is where a majority of your ATP is produced. Like I just said, oxygen is going to be the final electron acceptor. Hint, hint, that's a big concept here. Combines with hydrogen to form water, just like I said. Now, one thing I do want to go over here is how much ATP is produced per NADH and FADH2. So per NADH, because this is where we're going to have to do a little bit of math, and it's like fifth grade math, so don't get too worried here. NADH, for every NADH that your body produces, so we produce NADH and FADH2s in the first three steps. For every NADH, you're going to produce about 3 ATP. For every FADH2, you're going to produce around 2 ATP. The reason why I'm saying around and like these are rough numbers, it's not that whenever like we can watch it as a scientist, like we can like peer into the mitochondria and we can see an NADH drop off its hydrogen ions and the electrons and see 3 ATP be produced. We can do a lot of tests and simulations. Not simulations, sorry. Get a lot of data and crunch the numbers, and the data comes out to be about 3. There's no like specific way because how many hydrogen ions and how many electrons are being donated, and how many go through to make like there's a lot of just data out there that supports that it's around 3, but it's not like again it just makes automatically 3 every time NADH unbonds the electrons and hydrogen ions. So and again, every mitochondria might be a little bit different. If you have less ATP synthase or more ATP synthase, that could skew your numbers. But overall, the tons of data that scientists have accumulated, it's around 3 ATP. So what we're going to do next is we're going to look at how much ATP is produced in all at each step. So this is really important for you guys to be able to describe. So let's look at the 3 different phases, or I'm sorry, the 4 different phases, and look how much ADP, FADH2s, and NADHs are produced. So in glycolysis, we only produce 2 NADHs and 2 ATPs. So not a lot of ATP overall. The next step here, the prep step, we're only, again, producing 2 NADHs, but nothing else. So overall, the first 2 steps, we're still not making a lot of ATP. The Krebs cycle, also known as the citric acid cycle or the TCA cycle, we produce 6 NADHs, 2 FADH2s, and 2 ATPs. Now, at this point, again, we've only produced 4 ATP. Not a lot of ATP. Your cells need millions and millions every second. That's not a lot of ATP. It means you'd be eating constantly if this is all the ATP you would produce. However, if we add up all of the NADHs and FADH2s, you're going to see we produce 10 NADHs and 2 FADH2s. If we do the math, 3 times 10, because we have 10 NADHs and every NADH makes 3 ATP, we get 30 ATP. For the 2 FADH2s, we're getting 4 ATP, because again, 2 times 2 is 4. So if we add these 2 together, we're going to get 34. So if we add everything together, we get 2 ATP at glycolysis, none at the prep step, 2 at the Krebs cycle, and 34 at the electron transport chain. It's 38 ATP for every glucose. Now, again, this is not like 100%. Like I'm going to ask you like, oh, it's every time. Again, every person's going to be a little bit different. It's around 36 to 38. That's why I like to give that little leeway there. It's between 36 and 38. On a test, again, I won't say like, hey, is it 36 or 38? I'm going to say, is it 38, 100, 2, something like that. So you can identify 38 ATP it's around. If you remember back a couple sections ago, we talked about how the first step was anaerobic, which means it doesn't require oxygen, and the next 3 were aerobic. Now, I didn't talk about oxygen until electron transport chain. So you're probably thinking like, why are the prep step and Krebs cycle also aerobic, but if we didn't really talk about oxygen? So here's why, and this is again a great place where I can show you how different it is between like your ninth and 10th grade biology class and your AP class here. So ninth and 10th grade biology, I would just given you and said, hey, or giving you the information, hey, the first step is anaerobic, the next three are aerobic, and you just need oxygen for them. So I want you to be able to tell me why they're aerobic. So if there's no oxygen present, the electrons will be stuck in the electron transport chain. What that means is the NADH and FADH2s won't be able to drop off more electrons. It's like a bucket brigade where the first person, or I should say the last person, stops working and they just hold their bucket. Everyone else gets backed up and you can't get anything else through that chain. So what will happen is all the NADHs and FADH2s will become filled up, which means they can't go to the prep step, Krebs cycle, or really glycolysis. We'll talk about that here next section, and get more electrons. Let me show you what that kind of looks like here. So what happens here is you can see oxygen is going to take on those electrons from the electron transport chain. However, if you don't have oxygen, let's say you're going through a very vigorous exercise and you're breathing really hard, you don't have a lot of oxygen in your body, you're not going to have any oxygen to take on those electrons. So what happens is these electrons get backed up to a point where NADH can't give more electrons to the electron transport chain. Or FADH2, I should say that as well. So then these FADH2s and NADHs can't go back and work within glycolysis, the prep step, and Krebs cycle. So what happens is we don't get any more FADs or NADs. If we don't have any FADs or NADs, the prep step, Krebs cycle, and essentially the glycolysis, the first step, won't work. So this is why those next two steps, or I should say the prep step and the Krebs cycle and the electron transport chain don't work without oxygen. If there is oxygen, you can see these hydrogen ions will move through here. However, if there's not oxygen, we're not pushing hydrogen ions here. We're going to find an equilibrium between these two sides, and then we'll stop producing ATP. And that's a bad thing. If you don't have ATP, you can't do things like breathe, and you kind of need to breathe. So this is why the second, third, and fourth step are all aerobic. It's because this step specifically is aerobic and needs the oxygen. If this step doesn't occur, then the second and third step won't occur either. We'll talk about in the next section how the first step, glycolysis, can still occur without oxygen, and how it uses or how it kind of frees up NADH in a way.